Hello, I'm Zeynep Doğan.
I am YILMAZ. We analyze the Peto paradox with bioinformatic analysis. Cancer is the uncontrolled proliferation of cells due to errors in cells, in tissues and organs. We compare the genomes of organisms which is living in the polar regions with the human genome in the study of cancer. We have benefited from the field of bioinformatics in the comparisons analysis. Bioinformatics is a multidisciplinary field that provides an opportunity to drug and gene design.
We normally think that larger animals are more likely to get cancer. The reason we think like this is that because of that they have too many cells and these cells might get out of control and increase the risk of cancer. However, we see that large animals are less likely to get cancer than smaller animals. This situation is called the Peto paradox. In other words, there is no relationship between body size and cancer. Why large animals are less likely to get cancer? Decreasing in oncogenes, increasing in tumor suppressor genes, decreasing in metabolic rate, increasing in immune resistance, and hypertumor fermentation play a role among the reasons. The difference of our study from other studies. There is no exact cure for cancer. The bioinformatics field we use in our study is up to date. Supports laboratory studies, save cost and time, and enable the analysis of complex molecular structure. Lack of study on MSH2, CADM1, and OTCH1 proteins in Balaenoptera physalis, Balaenoptera acutostrata scamoni, and Balaenoptera musculus, species living in polar region.
In our study, we performed on tumor suppressor genes, which is one of the arguments of the Peto paradox. We handled cancer in 4 states. We identified cancer-associated tumor suppressor genes in the KEGG database. We took the sequence of MSH2, P53, NOTCH1, CADM1 proteins from the UniProt and NCBI databases and aligned them in the COLLAGE program. MSH2 plays a role in repairing DNA damage, while 95.50% similarity was observed between corrugated waste and humans. We found that there was 96.15% similarity between Milky Ways and humans. P53 is found in cell division checkpoints. When it encounters an irreparable error during cell division, it directs the cell to apoptosis to prevent cancer. The human P53 protein has 16.91% similarity compared to the blue wave. It is seen that there was a similarity of 24.09% with water bears, 86.53% with finback whales, and 86.27% with minke whales.
NOTCH1 is a proto-oncogene. Proto-oncogenes are prone to cancer, and when they mutate, they turn into oncogenes, that is, cancer cells. We found 88.63% similarity when we compared human NOTCH1 protein with minke whale. CADM1 protein is the protein responsible for metastasis in human. Human CADM1 protein has 98.19% similarity when it is compared to minke whale.
Adaptation developed by animals that are resident to cancer constitute important data in cancer research. Our study on tumor supersorgens, which are the components of the Peto paradox, gave us important results. In our alignment studies in the water whale, slot whale, minke whale, and blue whale, we determined that the human p53 protein also has many deletion and mutation sites. We determined that the mutation in which the p53 protein showed the least change was the water whale. The fact that the p53 protein changed too much in us shows that our probability of getting cancer may be higher. In our alignment study with humans, minke whales, and sloths, we detected a single deletion site and point mutation in humans. We determined that the MSH2 protein is highly conserved in all three organisms.
MOTCH1. While three deletion region of MOTC1 protein, which is associated with proto-oncogenes, were detected in humans, 10 regions were detected in minke whales. The loss of these regions in minke whale is thought to prevent the conversion of proto-oncogenes into oncogenes cancer genes. CADM1. Compared to human CADM1 protein with minke whales, 2 deletion sites were observed. This shows us that metastasis plays a role in regulation in human and minke whales in the same way.
When we look at the data which is supporting our study, it is the p53 protein with the most mutation and deletions. These studies show that our analysis is correct. Thank you for listening to us.